Welcome back to Enzyme Regulation, Control of Enzyme Activity. This time, we will discuss how the concentration of enzyme can be regulated at the RNA level. To get the most out of the videos, I recommend watching them in order. This is a reminder that this presentation is Dr. Johnson's intellectual property. In the early part of the 21st century, our understanding of the different roles of RNA in cells changed dramatically. Prior to this point, we believed that the main roles of mRNA were as mRNA and tRNA. The discovery that RNA could have regulatory functions in cells was a paradigm shift in our approach. There are several different types of RNA involved in interfering with translation of mRNA. Loosely, these types can be called RNAi and are comprised of microRNA, short interfering RNA, and picoRNA. These small RNA segments are used to stop or prevent translation of mRNA by interfering with ribosomal interaction with the mRNA. They tend to be important at different developmental stages of an organism. A different approach that is used by RNA is with riboswitches. These are metabolite-sensing RNA sequences that usually occur upstream of the operon for the metabolite. So far, they are only known in bacteria. We'll look at both of these in a little more detail on the following slides. MicroRNA forms imperfect hairpins or stem loop structures, while short interfering RNA are short segments of duplex RNA. Upon binding to the protein dicer, these RNA structures are cleaved into smaller duplex RNA segments. One of the RNA strands complexes with another protein and then base pairs with mRNA. The protein in the complex helps to stabilize the duplex. The overall effect of these duplexes bound to mRNA either represses translation, in the case of microRNA, or targets the mRNA for degradation, in the case of siRNA. Riboswitches are hairpin structures that occur near the Shine-Dalgarno, or ribosome binding site. The location and stability of the hairpin structures is altered by binding of a metabolite when it reaches sufficiently high concentrations. This is illustrated in Figure A. At low metabolite concentrations, the metabolite sensing region does not have significant binding of the metabolite, and the ribosome can bind to the mRNA and initiate translation. At high concentrations of the metabolite, the sensor region binds to the metabolite, forming a stem loop structure that prevents the ribosome from initiating translation. In Figure B, you can see the availability of the Shine-Dalgarno, allowing translation. Upon metabolite binding, there is a structural reorganization of the stem loop structures such that the Shine-Dalgarno is incorporated into a stem loop, which the ribosome is not capable of recognizing, so translation of the protein does not occur. This has been a very brief overview of regulation of enzyme production at the level of nucleic acids. If you want more detail, you should be able to find this in a modern molecular biology text. Next time, we'll see how the actual concentration of active enzyme can be altered.